Welcome to this explainer. We usually think of the eukaryotic cell as just this microscopic, kind of chaotic blob, right? Well, actually, scratch that. It's more like a bustling, incredibly organized metropolis. And right at the very center of this metropolis is a massive industrial complex known as the endoplasmic reticulum, or just the ER for short. Honestly, if you really want to understand how life operates at a fundamental level, you absolutely have to understand this factory.
Okay, let's take a quick look at the floor plan for our tour today. We're going to break this down into 5 key departments, starting with a quick overview of the cellular factory, then heading into the rough ER's assembly line, checking out the smooth ER's chemical plant, looking at some vital quality control and stress protocols, and finally seeing how all of this ties in to disease and biotechnology.
So let's kick things off with part 1, the cellular factory. This is our big picture view of the main manufacturing, processing, and storage hub. To really wrap your head around this, you've got to picture the ER as a sprawling, highly automated microfactory. But here's what makes it so incredibly unique. It's all one single continuous membrane. It's not just a bunch of disconnected little islands floating around. It's a physically connected infrastructure that is literally central to the eukaryotic cell survival, handling everything from building raw materials to managing cellular waste.
And there's a really stark functional divide between the two massive wings of our factory. You've got the rough side, which is absolutely packed with ribosomes and focuses almost entirely on protein synthesis and folding. Then you have the smooth side. This side completely lacks ribosomes, but it specializes heavily in lipid synthesis and detoxification. So they're part of the exact same network, but they have completely different job descriptions.
Let's move right into section 2, the rough ER assembly line, which is our dedicated protein production wing. We'll see exactly how it builds the proteins that keep you thriving. The manufacturing process here, it is strictly linear and highly organized. First up, ribosomes attached to the outside surface synthesize the raw protein. Once that's done, the protein moves into the internal space, the ER lumen, to start folding into its complex 3D structure. Next, the ER makes some crucial early modifications, like end-linked glycosylation. And finally, the processed protein gets packed up into transport vesicles and shipped out to the Golgi apparatus for final sorting. It's an assembly line in the truest sense. Now, what's totally wild is the sheer variety of crucial products this assembly line cranks out. The rough ER isn't just making generic filler. Nope, it is producing secretory proteins like the insulin that manages your blood sugar and the antibodies that literally fight off infections. Plus, it's manufacturing the membrane receptors that let your cells communicate and even digestive enzymes bound for lysosomes.
But proteins don't just magically snap into the right shape on their own, right? Enter our molecular chaperones. You can think of them as the dedicated floor managers of the ER. Their one and only job is to assist those newly synthesized proteins, making absolutely sure that every single one folds into its precise, functional, three-dimensional shape before it is allowed to leave the factory. Without them, the whole assembly line would just grind to a halt with tangled masses of useless proteins.
All right, leaving the protein line, we step into section 3, the smooth ER, chemical plant. This wing is all about lipids, steroids, and detox. So the big takeaway here is that the smooth ER is indispensable for life in ways you might not initially expect. Sure, it produces essential membrane components like phospholipids and cholesterol, but beyond that, in specialized cells, it actually teams up directly with your mitochondria to synthesize crucial steroid hormones. We're talking cortisol, estrogen, testosterone, you name it. It is an absolute chemical powerhouse. And if you've ever wondered how on earth your body clears out alcohol, medications, or foreign environmental compounds, well, you can thank this specific department, especially in your liver cells. The smooth ER uses a highly specialized group of enzymes called cytochrome P450. These enzymes actively metabolize toxins, fundamentally altering their chemical structure just to make them easier for your body to safely eliminate. This is where we see just how incredibly adaptable the smooth ER really is. In an everyday, regular cell, it acts as a general reservoir for calcium, quietly regulating things like cell signaling and secretion. But in your muscle cells, it evolves into a highly specialized subtype called the sarcoplasmic reticulum. In there, the controlled release of calcium is exactly what triggers literally every physical movement you make, from a single heartbeat to an all-out sprint. But hey, no factory is perfect, right? That brings us to section 4. Quality control and stress, covering the factory's crucial emergency protocols.
It really is a fascinating biological question. What actually happens when this microscopic factory inevitably produces a bunch of defective, misfolded parts? How does it handle a massive buildup without completely shutting down, or worse, destroying the larger organism? Well, the very first line of defense is a system known as ERAD. Think of ER-associated degradation as the factory's automated recycling department. When the ER checks out a newly synthesized protein and finds a misfolded one that those chaperones just can't fix, ERAD kicks in. It removes the bad protein from the ER entirely and ships it off to the proteasome, where it's safely broken down and destroyed. But sometimes the system just gets totally overwhelmed. If too many misfolded proteins pile up, the cell experiences what we call ER stress. To survive, it triggers the unfolded protein response, the UPR. This initiates 4 distinct emergency steps. It immediately hits the brakes on new protein production. It ramps up fold-in capacity, accelerates the removal of the junk proteins, and tries desperately to restore normal function. But a quick warning, if this severe stress persists and the ER simply cannot be saved, the cell pulls the plug. It initiates apoptosis, which is basically a self-destruct sequence, just to protect the rest of the body. And it goes way beyond just clearing out a bad protein here or there. The ER is unbelievably resourceful on a macro scale. It's deeply involved in a process called autophagy. Get this, the ER actually donates its own membranes to help form autophagic structures. These act like cellular garbage bags that swallow up and recycle much larger damaged cellular components, keeping the whole cell healthy and efficient. Let's step out of the microscopic world for our final part, Section 5, Disease and Biotechnology. This is where we see the ER's impact in real-world medicine. Because here's the thing, persistent ER stress and defective quality control, they aren't just microscopic hiccups. They are central to major global health crises. When the ER's normal functions break down, it directly contributes to devastating conditions like neurodegenerative diseases, diabetes, various cancers, and metabolic disorders. And that is exactly why the ER is such a massive focal point of modern medical research. But on the flip side, there is incredible potential for biotechnology. Scientists are deeply engaged in protein engineering right now. By understanding exactly how the ER folds and secretes proteins, bioengineers can modify these systems. We are literally hacking into these microscopic assembly lines to mass-produce complex, properly folded, life-saving biopharmaceuticals, stuff like therapeutic antibodies, enzymes, and hormones. It's a real game changer. As the research points out, there is a massive opportunity right in front of us. If we can just deeply understand and actually chemically control the ER stress responses and its quality check mechanisms, we unlock a completely new, highly targeted frontier for drug discovery. And so that brings us to the ultimate question, leaving you with something to really ponder. Just how much untapped potential is still hidden within this bustling, incredibly complex network? As we continue to decode the secrets of the ER, we aren't just learning how cells work, we are quite literally learning how to rewrite the factory code to cure the diseases of tomorrow. Thanks so much for joining me on this tour of the cell's ultimate microfactory.